What if doctors could diagnose cancer patients earlier by visually tracking when anti-tumor genes are not functioning properly? Currently, we do not have ways to diagnose cancers early, and this is a problem because the longer a patient waits before treatment, the further the cancer could progress in the body. One way to address this issue is by adding some sort of reporter gene, like a fluorescent protein in the patient's DNA. This can be done through molecular cloning. This process involves cutting the gene of interest out of the native DNA genes with restriction enzymes, which are like scissors. The gene of interest can then be inserted into a plasmid or bacterial DNA genome using ligase enzymes which act like glue. Then this plasmid is introduced into the bacteria to produce many copies of the specific gene of interest. A good way to think of how the system works is to think of the body as a car. In a car, the catalytic converter is used to reduce the toxic components in the gas emitted by the vehicle. Similarly, tumor suppressor genes in your body regulate cell growth to prevent uncontrollable cell growth, otherwise known as a tumor. However, when a catalytic converter malfunctions, carbon monoxide, a poisonous gas, can leak into the car. When the tumor suppressor genes malfunction, cancerous tumors can develop. In cars, there are check engine lights to alert the driver of the problem. Just like the check engine light in a car, scientists can utilize reporter genes, such as fluorescent proteins, to reveal the loss of expression from tumor suppressor genes. Doctors can reveal any issues by shining a light on patient cells and looking for cells that glow from the fluorescent protein. So through using the molecular cloning process to link GFP with tumor suppressor genes, doctors could visually track the expression of these genes, which could reveal the early development of cancer in a patient. In conclusion, scientists are beginning to use this on human cells, leading to further research and expanding the possibilities this process could play in furthering the medical field.